Welcome to the Biology of GIST, what every patient needs to know. Please remember that information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild, the Vice President of Program Services, and I am your moderator for today. I would like to introduce you to our presenter today, David Josephy. He's a longtime friend of the LifeRaft group. His background is that he's a professor of molecular and cellular biology at the University of Guelph, where he has taught for more than 35 years. He has been president of GIST Sarcoma LifeRaft Group Canada since its inception in 2008. David is a member of the LRG science team and collaborates with GIST patient support groups around the world. His research interests are in the field of molecular toxicology, and he's particularly interested in the scientific basis for development of new small molecule drugs for GIST. We're happy that David has joined us today, and I'm handing this off to David to begin.
Thank you, Sarah, and thank you everybody for dialing in or logging on. So just to repeat the disclaimer that Sarah gave you already, I'm not a physician, not a medical doctor. I'm a biochemist, so this talk is focused more on the basic biology of GIST rather than the medical aspects, although of course those two subjects are closely intertwined. We begin by pointing out to you that we have, about a year ago, we put together a top 10 list of recommendations and advice for newly diagnosed patients. That's available on our website of LifeRaft Group Canada, and if anybody's interested, just contact Sarah or me afterwards, and we can send that along to you. So I want to talk about a variety of topics today related to the biology of GIST, a little bit about what GIST is as a disease and what causes it, the simple answer to that question being that we don't know, say a little bit about the diagnosis and treatment of GIST. Then I want to turn to the biology, the underlying biology of GIST from a cellular and molecular point of view. We will talk about KIT and PDGFRA. These are the names of two proteins that are closely involved in most cases of GIST. We'll say a little bit about the TKI or tyrosine kinase inhibitor drugs, such as imatinib and Gleevec. Let's see, advance here. Sorry. Okay, so what causes GIST? GIST can be an inherited disease due to mutations that are passed in the family from one generation to another. This is very rare. There are some cases in the U.S. that have been identified, but we're talking about really just a very small number of families that are known to have inherited GIST worldwide. It's exceedingly rare. GIST is rare, but it's not exceedingly rare. So the vast majority of GIST cases are what we would call sporadic GISTs. That is, they occur in individuals without any family members being affected and without the possibility of passing that risk or that disease on to children. So almost all GISTs are sporadic. I would say very simply, we know of, when a newly diagnosed patient, inevitably the first thing people think is, what have I done? How did I bring this disease upon myself? Well, you didn't. We know of no environmental or dietary or lifestyle or occupational or geographical causes of GIST. There is no known such association. The rate is pretty much the same wherever you look around the world. And I would say furthermore that if there were any major causative risk factors like cigarette smoking, for example, we would have noticed that a long time ago. So we know of no avoidable causes of GIST. It really does strike at random. So what is GIST? Let's begin by recognizing that the human body or any animal's body is made up of an enormous number, hundreds and hundreds of different types of cells. And I've just shown, more or less at random, a few of these different cell types in the human body, just to illustrate how very different they are. So blood cells at the top left, hepatocytes, the major cell type in the liver, example of muscle cells in the heart, fat tissue. You can see that all of these cells look very different, even though they have the same genome, the same genes, but they look and behave very differently. A cancer can begin in almost any cell type in the body. And a fundamental point to understand about cancer is that it is the type of cell from which a cancer develops that defines the cancer, its diagnosis, its treatment, its biology. It's absolutely crucial that the cell type of origin of a cancer has to be correctly identified. That's usually done by looking at a specimen of the tumor taken at the time of surgery. It is that specimen is then passed on to a specialist doctor, a pathologist, and ultimately it is the pathologist who writes the report that gives his or her best estimate, best evaluation of what the cell type is, and therefore the nature of the cancer. We all hear a lot about cancer. We all know people affected by cancer. We all read about cancer in the media. Most of the time when you hear about cancers, you're hearing about a class of cancers called carcinomas. These are the great majority of cancers. Carcinomas arise in lining tissues that line either the external or internal linings of the body. And so carcinomas include most of the common cancers that you will hear about day to day. Skin cancer, colon cancer, lung cancer, breast cancer. GIST does not belong to that group of diseases. It's a different class of cancers called sarcomas. Sarcomas do not arise from these epithelial lining tissues. They arise from a different kind of tissue, cells that are part of connective tissues, such as the blood vessels, cartilage, bone, and other internal connective tissues in the body. Sarcomas are rare among cancers. They're not unknown, but they're rare. Something like 1% of all cancers are sarcomas. That's why when you hear people talk about cancer, they're almost always talking about carcinomas. Sarcomas are just very relatively rare. At the bottom of this slide, I've shown just a cartoon cross-section of the lining of the GI tract, the stomach in this case, to make the point that carcinomas, which happen in the GI tract, colon, stomach, esophagus, carcinomas begin with cells that are on the lining of the GI tract, which is the inside surface of the stomach, for example, recognizing that that's really the outside of the body because the GI tract is really external to the body, even though it runs through the middle of the body. GISTs, on the other hand, as sarcomas, start on the outside, actually in the muscular wall that surrounds the gastrointestinal tract. So they are sarcomas rather than carcinomas. In fact, we now understand which particular cell type is the origin of GIST. It's a class of cells that none of us paid any attention to or heard about until we learned about GIST. These are cells that were first recognized by a famous in the biology community, a famous Spanish histologist or biologist named Cajal, and he identified these cells, and they're known as the ICCs, or the interstitial cells of Cajal. These cells are neuromuscular, as we say, neuromuscular cells that surround the GI tract all the way along the GI tract from the esophagus right to the anus. And what do these cells do? Well, to digest our food, how does the food move along the GI tract, through the stomach, through the intestines? The food is moved by waves of muscular contraction of muscles that surround the GI tract. We call that movement peristalsis. These are waves of contraction that push food along the GI tract. And to have these coordinated waves of muscle action, there has to be a pacemaker. There has to be a neural type cell that sends out the regular pulsed signals telling the muscle to contract. And those cells that do that task are called the ICCs, the interstitial cells of Cajal. So GISTs begin either with the ICCs themselves, the interstitial cells, or perhaps they begin with a precursor cell to the ICC. That's still not clear. But effectively, we can regard them as tumors that originate in these neuromuscular cells, the ICCs along the GI tract. And that then explains why GISTs can and are found occurring anywhere along the GI tract, the gastrointestinal tract, all the way, as shown in this picture, all the way from the esophagus to the rectum. Most commonly in the stomach, but potentially at any site along the GI tract. Very fundamental point to appreciate is that because the cell type, the ICCs, are distributed all the way along the GI tract, any of these tumors is a GIST, regardless of whether it's developed in the stomach or the colon or the esophagus. They're all GISTs because they begin with the same cell type. Now, there are biological differences between GISTs in the stomach and GISTs in the colon, for example. There are some differences in behavior. But nevertheless, fundamentally, it's all the same disease. Okay, and now I'll emphasize this again because it's an important fundamental fact, and it does sometimes cause confusion. So just as in my classes on campus, when there's something important to say, I will say it more than once. So first of all, a GIST that starts in the stomach is a GIST. We hear all the time people saying, I've developed a rare stomach cancer, or journalists talking about GIST and saying it's a rare stomach cancer. It really isn't. So when people say a stomach cancer, they're normally referring to the carcinomas that begin in the stomach, rather common cancers. Usually these are what we call adenocarcinomas. So a GIST is a GIST. It's not a stomach cancer. And when people talk about colon cancer, which is a very common cancer, one of the most common, they're usually talking about carcinomas, colorectal carcinomas, and even if it just begins in the colon, it's a GIST. It's not a colon cancer. Let's not call it colon cancer. That would only cause confusion. It's true of all cancers, including GISTs, that they can metastasize. That term means that the original primary tumor that developed, for example, in the outside the stomach, can shed cells that then travel either locally or through the bloodstream to different sites in the body. They attach at those sites and they begin to grow. GISTs in particular tend to spread locally into the peritoneal cavity, the abdominal cavity, and they can also spread to the liver, the lung, even to the brain. I've highlighted this in red because it's another important point that is often a cause of confusion. Metastases, and we often call those METs for short, wherever a MET arises, if it's a GIST MET, it has the biological properties of GIST. Metastases have the same biological properties as the primary tumor. I like to say sometimes when I'm at a meeting in Canada or in England, in the USA or in England, I say I'm a Canadian. The fact that I've traveled to a new location in the US or in England or in Australia, it doesn't change my fundamental property of being a Canadian. And in the same way, if a GIST cell travels and regrows in the liver, it's still a GIST cell. So GIST mets in the liver or the lung or wherever, they're still GISTs. They have to be treated as GISTs. So again, we quite often hear people say, I used to have GIST, but now my doctor told me I have liver cancer. But in most cases when we hear that, it's simply a confusion. Actually, the patient still has GIST, but the GIST is now metastasized to the liver. It's not a liver cancer. A liver cancer would be a cancer that began and arose from liver cells. I hope I've made that point clear now. Now to turn to the biology and the cell science of GIST. Before 1998, that was the dark ages as far as GIST is concerned. Almost nothing was understood about the origin of the disease. A huge improvement, a huge revolution in our understanding began with a publication in 1998 by a group from Japan, notably the photos are shown here, Dr. Kitamura and Dr. Hirota. This discovery was the result of many decades of research leading up to the discovery. I won't go over all that. But what these doctors discovered is that, first of all, that GIST cells, tumor cells, express, that is, produce, a particular protein called KIT, K-I-T. The name actually comes, it sounds like kitten, and it actually comes from kitten because this gene was first identified in a cat virus, a feline virus, but we needn't worry about that. It's really a historical fact. So this protein called KIT is expressed in GIST cells. It's also expressed in ICCs, which shows you the relationship between ICC cells and GIST. But importantly, very few, there are some, but very few other cells in the adult body express this protein. Secondly, these Japanese researchers discovered that in most cases of GIST, although KIT is produced, it's actually an abnormal form of the KIT protein resulting from a mutation or a genetic change that has occurred in the gene that encodes KIT, the KIT gene. So I'm going to spend a little while now just exploring and elaborating on this, on what I've just said, to try and make it more transparent and clear. But let me also say before I leave this slide that everything we knew about GIST before 1998, pretty much everything is obsolete. So there's really no point worrying about what we knew about the treatment or diagnosis or biology of GIST before 1998. It's essentially swept aside by this discovery that opened a new understanding of the disease. And let me update that slide because it's now 2019. We've moved on a couple of decades beyond that original discovery. So let me just elaborate a little bit on what I just said in the 2019 context. It turns out that we now know that GIST, which we always thought of as being a single disease, you might really regard it as being an umbrella diagnosis. Actually, there are several quite different forms of GIST which have now been teased apart genetically by genetic and molecular analysis. So what we thought was really one disease is, in a sense, a group of different but related diseases. So I said a moment ago, the 1998 discovery that most GISTs have a mutation in the KIT gene, and that's still true, but not all. It's about 3/4, roughly, of GISTs that are KIT mutant. There are about 25% that are not KIT mutant. I'll talk a little bit about those cases later on. In fact, there are about 10% that are so-called wild-type GISTs. Many of you will have heard that term, wild-type GIST. Wild-type is what the term a geneticist uses to say the gene that has not been, a normal gene that has not been mutated. We're learning more about wild-type disease, GIST, every year. It's now quite clear that wild-type GIST is really a distinct disease, quite different in its diagnosis and treatment in its biology and treatment compared to KIT or PDGFRA mutant tests. So I'll elaborate on that as we go along. Okay, back to the beginning, and this is a slide that I could be showing, that I do show in the course that I'm teaching at the university right now. So let's go back to the basics of molecular biology. Each of our cells contains a genome, our own genome. That genome is built out of the molecule DNA. That's the molecule that makes up the genetic code in the chromosomes. The gene, the DNA, provides the information, that is, as we say, the blueprint or the code that tells the cell how to make proteins. There are something like 30,000 or so different human proteins, and each encoded by a different gene. Here I'm oversimplifying a little bit, but not too much. So at the top of the picture, we see the famous double helix of DNA and a little bit of DNA sequence shown to the right. DNA is used to assemble a molecule called RNA, and RNA in turn is used to assemble proteins. If you want to know the details, come to the University of Guelph and listen to my course for the next 6 weeks or so. But we don't have that much time this morning. So that's the essence of molecular biology, and that's the background we need to understand the role of KIT and KIT mutations. So what is KIT? By the way, it's sometimes called c-KIT, and it's sometimes called CD117, which is an obscure kind of telephone number of a name. But that doesn't matter. It's all the same protein. These are just old-fashioned synonyms for it. So what is KIT? Well, KIT is a specific protein. It's one of those 30,000 or so different human proteins. And as I've mentioned earlier, it's made, that is, we say expressed or produced, by only a very few types of adult cells. That includes the ICCs, and it includes GISTs. Almost all GISTs. More than 95% of GISTs express KIT. In fact, because GISTs almost always express KIT, this is the most important way that the pathologist can make sure that a tumor really is a GIST. It's the essential step in diagnosis. The pathologist needs to know whether the tumor cells express this protein, KIT. That's done by a specialized technique with the long name immunohistochemistry. I'm not going to go into detail about that today, but this technique, which is established in any pathology lab, it's a staining technique using an antibody, and it allows the pathologist to determine whether KIT is being expressed by the cells in the tumor sample. And if it is expressing KIT, then that's very strong evidence, almost conclusive evidence, that the tumor really is a GIST. And by the way, the protein that's expressed may be a mutated version of KIT due to a mutation in the KIT protein, in the KIT gene, or it could be the wild type, normal version of the KIT protein. That happens too, but whether it's mutant or wild type, it is expressed by the cells. So I've referred many times now to mutations. What are mutations? Mutations are simply changes in the DNA sequence that encodes the protein. Mutations occur at random every time a cell divides. Of course, natural selection, as Darwin taught us, natural selection will allow cells carrying certain mutations to survive and grow, to get a growth advantage, and to grow preferentially. Other mutations will be deleterious or damaging to a cell. And so although mutations occur randomly, the consequences of those mutations are not random. Okay. KIT mutations. I've said that the KIT protein is a protein. In fact, it's an enzyme, as we say in biochemistry. That is, it's a molecule, a protein molecule that catalyzes particular chemical reactions. And it is belongs to a large class of human enzymes and proteins, which we call tyrosine kinases. I don't really have time to elaborate on all of that in this short presentation, but tyrosine kinases are enzymes that act on other enzymes. They're proteins that modify other proteins. This is part of a process which we call signal transduction, which really represents the way in which the cell responds to the environment, responds to external signals. As I mentioned already, most GISTs, not all, but most GISTs, carry a mutation in the KIT gene. The consequence is that they produce an unusual form of the KIT protein, and that mutated KIT protein drives the growth of the tumor cells. So the KIT gene is one of many genes that have been identified and put into the class of oncogenes. Onco for cancer, onco meaning cancer, cancer genes. So oncogenes are genes, and there are many known, that encode a protein which, when it has a mutation, can send an instruction to the cell to carry on dividing, whether it should or should not. A good analogy for this is it's like having the gas pedal in your car stuck to the floor. So gas keeps on being supplied to the engine even when you don't want it to be. And so when the KIT gene is mutated, it acts as a driver, as we say, a driver mutation that keeps telling the GIST cells to divide and proliferate, even though they should not be doing so. Let me emphasize again that in almost all cases, with the exception of those very, very rare familial cases, the KIT mutations in GIST are almost always, as we say, somatic. That is, they occur in body cells, not in germline cells, eggs or sperm. So consequently, these are mutations that occurred in a patient's body during development or possibly during adulthood. They do not affect the germ cells, and therefore they cannot be passed on to a patient's children. The only exception to that rule are the exceedingly rare cases of familial GIST. Now, on to talk a little bit about the diversity of mutations in GIST. As I said a moment ago, the KIT gene mutations, act as drivers of the growth of GIST cells. In fact, those mutations can occur at many different points within the KIT gene and therefore within the KIT protein. More commonly in certain places than others, but at many places throughout the protein. Furthermore, we now know in research that was performed since 1998, since the original discovery of KIT and its involvement in GIST, we now know that there are several other genes that can also undergo a mutation that drives the GIST. The next one to be discovered after KIT was a gene called PDGFRA. And more recently, mutations have been discovered in occasionally in GIST, in other genes with names like SDH, BRAF, tract, and more. All of these mutations occur in GISTs, but we understand biologically that the mutation matters. The site of the mutation has an important influence on the prognosis of the development of the disease and on how the GIST will respond to different drugs. An important example of this, as we now understand, is that a significant number of GISTs, and that includes the ones that we call wild type or sometimes quadruple wild type. It includes quite a large subcategory of GISTs, the so-called PDGFRA D842V mutations, which I'll explain a little bit later. GISTs that are driven by these particular mutations almost certainly do not respond to imatinib or Gleevec. Nevertheless, many patients with these mutations are still being prescribed and taking imatinib. We think that that's wrong. It's unnecessarily exposing those patients to the side effects of a drug that really isn't doing them any benefit. So we've been saying for more than a decade that all new GIST cases should be provided with mutational testing of the tumor cell DNA to identify the nature of the driver mutation. And it's taken a long time to get this implemented. There are a few countries now, Belgium is actually one example, where this is already implemented, and every new GIST case is being studied by mutational testing. Germany, pretty much every new patient is being given mutational testing. Unfortunately, in the USA and Canada, the rate of mutational testing is still appallingly low, maybe only 25% or so of cases that are getting mutational testing. We're working very hard to try and increase that number because we think every new case should get mutational testing. That's what it says here. So all GIST patients should get mutational testing. That is to say, all doctors should be ordering it to be done. The only country so far where it's being done on every case is Belgium. And in the U.S., it's still being highly underused. This is I've shown you here a paper that came out just last year by a group of doctors treating GIST in Washington, D.C. And even in Washington, D.C., in the nation's capital, even there, they're complaining that mutational analysis is being highly underused. It's not being done as often as it should be. All right. So I said earlier that a mutation is a change in the DNA sequence encoding a protein. Many of you will have heard of terms like exon 9 and exon 11 KIT mutations. So what does that mean? What is an exon, E-X-O-N? So this is a specific term used in molecular biology. Almost all proteins, and therefore the genes that encode the sequences of those proteins, have what we could call a modular structure. It's like they're put together out of a kit of component pieces that are assembled together. Maybe 5 or 6 or 8 different parts of the protein which we call domains. So a domain is like a functional piece of a protein that these domains are assembled together to make the full protein. And typically, each domain is encoded by a separate section of a gene which we call an exon. Now, to explain that in more detail, yes, we'd have to go on to a whole course in molecular biology. But essentially, an exon is a piece of a gene that corresponds to a functional piece of a protein. So on this slide, I'm showing you a pie chart that represents the distribution of mutations in GIST. So to explain this, I said a moment ago that we think that all GIST cases should undergo mutational testing, DNA analysis. We aren't there yet, but nevertheless, we already have a database of thousands of GIST cases that have been studied by mutational testing. And so we can look at that database and ask, how common is each particular kind of mutation in GIST? What percentage of GIST patients have these mutations? And this kind of analysis has been done quite a few times now in quite a few different countries. I'm showing you one particular example of that kind of analysis, but it's representative of what we see all around the world. So in this pie chart, you can see there's a very large blue or cyan-colored segment on the bottom right, and that's labeled KIT exon 11. Something like 60%, more than half of all GISTs, when we study their mutations, we find that there's a mutation in one particular part of the KIT protein and gene, which we call exon 11. At the top right, somewhere around the 1 o'clock position, there's a dark blue pie labeled KIT exon 9. These are something like 10% of all GISTs. That's again a mutation in KIT, but occurring in a different part of the KIT protein. corresponding to exon 9. These, as many of you may know, are biologically somewhat different from the KIT exon 11 mutations. Typically, these patients are best treated with higher doses of imatinib relative to the exon 11 case. Now, if we move over to about 10 o'clock on this pie chart, you can see a violet-colored segment, a large segment. It's about 14% on this chart, PDGFRA exon 18. This is primarily made up of one particular mutation, the so-called D842V mutation, in a gene that is not KIT, but a closely related gene called PDGFRA. There's also a red segment on this pie chart, somewhere up around 11 o'clock. These are the so-called wild-type GISTs. As I said at the bottom of the slide, wild-type is a bit hard to understand that terminology. To a geneticist, wild-type really just means we don't see any mutation. So these are cases where no mutation has been identified, and we've ruled out any mutation in KIT or PDGFRA. Those genes were sequenced and they were wild-type. So we call that a wild-type GIST. It's a little bit misleading. Most of these cases probably do have mutations, but they're mutations in genes other than KIT and PDGFRA, in many cases mutations that we have not yet identified. Gradually, that red pie on the pie chart is shrinking as we identify more and more of these very rare mutations in occasional GISTs. For example, some of you have heard about SDH-deficient GIST due to mutations in a gene called SDH, and that would be a small slice, a component of that red pie on the pie chart. So what does this terminology mean that you will hear with regard to mutations? So, for example, you hear about KIT V560D. What does that mean? That's a mutation found in some GISTs. We call it a missense mutation. The number 560 refers to the 560th amino acid in the KIT protein. So proteins are long chains of amino acids, components of proteins, hundreds of building blocks, and one particular building block, number 560, has changed from the wild type, represented by the letter V, which stands for an amino acid called valine. It's been mutated to a different residue, an aspartic acid residue, represented by the code D. So V560D means residue 560 is changed from valine to an aspartic acid. Another kind of terminology you may see, quite a common mutation in KIT, KIT W557K558DEL. So what's that? That's a deletion mutation. In this case, two of the amino acid building blocks in the chain are just missing due to a mutation in the DNA, a deletion mutation. So two of those amino acids are just absent. And I think I show you that schematically on the next picture. So if you think of a protein as being a long chain of amino acids, a long linear chain like the successive cars on a train, if you think of the protein as being a long chain of amino acids, there are 20 different amino acids found in proteins. Each one has a symbol, a one-letter symbol, A, B, C, D, in this case E, P, Q, and so on. So what we're looking at here is just a little section out of the hundreds of amino acids in the KIT protein. This is just a little section of exon 11, beginning at residue 554, which happens to be a glutamic acid represented by E, and then the next 10 or so amino acids in the protein. So the missense mutation, V560D, is represented by the middle picture in this set of pictures, and you can see that one of those valine residues, number 560, has changed to a different residue, the green box representing the aspartic acid. The deletion mutation is represented at the bottom, and you can see that the W and the K residues are missing, And now we have a slightly shorter protein that's missing these two amino acids. A few more words about wild-type GIST then. The major target genes in GIST, as we've seen, are KIT and PTGFRA, but about 10% of mutations do not have, 10% of GISTs do not have mutations in those genes. We call those wild-type GISTs. As it says in red at the bottom, we've generally found that wild-type GIST is quite a different disease. It's still GIST, but a different category of GIST. It tends to affect younger patients. It tends to affect women more than men, girls more than boys. It tends to be, as we say, indolent or slow-growing rather than an aggressive tumor. And it, as far as we know, it really doesn't respond to imatinib or other TKI drugs. I don't want to be dogmatic about that because there may be some cases that do respond, but without doubt, overall, the response of wild-type GIST to imatinib is much poorer than for KIT-mutated GIST. SDH-deficient GIST, we don't have time to talk about the details, but this is a subcategory of wild-type GIST. The first publication recognizing SDH-deficient GIST was in 2011. It's only recently discovered. These so-called wild-type GISTs actually have a mutation in the gene for an enzyme called succinate dehydrogenase. If you've been through a biochemistry course, you might remember that that's a component of the so-called citric acid cycle, or Krebs cycle. We don't yet know enough about this disease, this type of GIST, but the research in this area is moving forward very quickly. At the Miami Life Fest last year, 2018 in Miami, organized by LifeRaft Group, there was a major international workshop focusing on SDH-deficient GIST, and some of the participants in that workshop are shown in this picture, including Sarah at the far right side. In the remaining few minutes, I guess I take about another 5 minutes. In the remaining few minutes, let me talk about the targeted drugs for treating GIST. This development was a remarkable breakthrough in GIST treatment around the year 2000. It actually came from a very unexpected direction. It came from the development of drugs for treating what appeared to be a totally different disease, a leukemia or blood cancer called chronic myelogenous leukemia, apparently completely unrelated disease, and yet it turned out that the driver mutation is in a related gene called ABL, another tyrosine kinase gene. And these doctors and scientists shown on this picture, notably Brian Drucker from Oregon, devoted themselves to trying to find a targeted therapy to treat CML. They discovered it in this drug called imatinib. It was an enormous breakthrough in cancer research. So as I say at the bottom of the slide, the Nobel Committee needs to wake up and give these folks the Nobel Prize because it was that important a discovery. It had apparently nothing to do with GIST, and yet it turned out to have everything to do with GIST. So the drug that they developed in collaboration with Novartis Pharmaceuticals, the breakthrough drug for GIST, originally called STI-571, now known as imatinib or brand name Gleevec. This is an inhibitor of this ABL tyrosine kinase. It's transformed the treatment of chronic myelogenous leukemia. It's also transformed the treatment of GIST. So it turns out that this tyrosine kinase that drives CML leukemia is quite closely related to KIT and PTGFRA, the tyrosine kinases that drive GIST. And so doctors made the leap of faith of thinking that maybe the drug that worked for CML would work for GIST, and it turned out that it did spectacularly well. So we have a first generation of drugs for treating what I will call the first generation of drugs for treating GIST. These include drugs many of you have heard of or and are taking, imatinib or Gleevec, sunitinib or Sutent, regorafenib, or so-called Stivarga. Bear in mind that none of these drugs were actually developed specifically for GIST. They were mainly developed for CML leukemia and other cancers, but they turned out to work pretty well for GIST. Now, around 2019, a second generation of drugs, what we can call bespoke or tailor-made GIST drugs, is, we hope, now arriving. So we hope for a further great improvement in GIST therapy because of these new drugs. This is just a tree. It's a family tree of proteins, in particular tyrosine kinases. And I'll just show you this to indicate that KIT and PTGFRA are on nearby branches. They're very closely related proteins and genes. ABL, the drug that the protein that is expressed in leukemia and for which imatinib was developed, that's quite far away on this family tree. It's still related, but it's a distant cousin, whereas KIT and PTGFRA are very close cousins. What do these drugs do? I'm getting towards the end of my talk now, so let me just say a few words about what these drugs like imatinib do. They are inhibitors of the activity of the KIT protein. So KIT carries out specific chemical reactions. Imatinib is like putting glue in the lock and preventing it from turning. So imatinib is a drug that inhibits the activity of KIT, and consequently it stops the GIST cells from dividing. Just a word about the naming convention. You'll hear about monoclonal antibodies like bevacizumab and cetuximab, and you'll hear about small molecule TKIs like imatinib and dasatinib. If it ends in ib, it's a small molecule, it's a pill that you take by mouth that inhibits the tyrosine kinase. Imatinib has been a tremendous breakthrough drug for GIST, but we still need more drugs. Some GISTs never respond to imatinib. Many GISTs respond to imatinib initially, maybe for many years, but over time the tumors tend to become resistant to imatinib due to additional mutations, and therefore we need to have additional drugs. So I've already mentioned these three. I won't go over this again. The three, which are now the first, second, and third line drugs for GIST, imatinib, sunitinib, regorafenib, which have been with us for more than a decade or so. Let me just conclude by mentioning some of these new drugs that we hope that are now in clinical trials. There are many clinical trials underway for GIST, various kinds of clinical trials, but among those trials are trials of new drugs which may be improved, as I say, bespoke drugs that are targeted to particular GIST driver mutations. And some of the most promising of these are a drug called larotrectinib being developed by Loxo and Bayer. This is a spectacularly good drug, but unfortunately it only targets one particular mutation called NTRK, and that's a very rare mutation in GIST, maybe much less than 1% of GIST patients. But if we can identify those patients, this drug looks great for those rare patients. Another drug you will have heard about quite a bit probably, developed by the Blueprint Company in Massachusetts. It was called Blue 285. Now it's got the name avapritinib. This was developed particularly to target those PTGFRA D842V mutations. And this seems to be a very promising drug in clinical trials. And finally, one more I'll mention, developed by the Deciphera Company, now called ripretinib. And this also appears to be useful for many KIT mutations. So we're really hopeful that these new drugs coming along, likely to be approved very soon by the FDA and other regulatory agencies, we hope there's going to be a further major improvement in GIST therapy. And there we are. I've got managed to end with about 10 minutes to go, so I'll leave it at that point.
Great.
And Sarah, are we now going to take some questions? I saw them popping up on the side of my screen, but I haven't had a chance to look at them yet.
Yes, I will go ahead and read them to you. Thank you so much, David, for your presentation.
Thanks.
So the first question, how long should patients continue the use of imatinib for exon 9 and exon 11 GIST for adjuvant treatment?
Yeah, so that's a tremendously important question, which of course I can't answer. Nobody really can answer that question yet. Let me just point out for those who don't follow the question, adjuvant imatinib, this is a particular use of the drug. So if patients have a localized tumor that's been successfully removed by surgery, after that point, there's no evidence that they even have GIST. There's no evidence of disease. But we know there's always a risk that a few cells were missed. Maybe a few cells were circulating in the blood. Eventually they could come back and the disease could recur. So it's been found that it is very advantageous in many cases to treat those patients with imatinib as almost as if they had GIST, even though there's no evidence of GIST. And we call that adjuvant treatment. Typically, this is done for up to 3 years. We would really like to know whether that period should be extended beyond 3 years. My guess is it probably should, but it's really a matter for an individual. There's no medical consensus as to how long you should continue adjuvant GIST. So this is really a question that has to be handled on a case-by-case basis by an expert oncologist who's really familiar with GIST treatment.
Great, thank you. If anybody has any questions, I know some people are raising their hands. Please type them into the question and answer window so that I can go ahead and ask them to David. So the next question is, I've noticed a wide range in mitotic rates between patients, some very low and others exceedingly high. What causes these differences in mitotic rates, and do subsequent tumors maintain these rates upon reoccurrence?
Yeah, another really good question. So mutation, mitotic rate, this is an aspect of the diagnosis that I left out of the talk for sake of brevity. So the pathologist, besides doing the staining tests that we discussed, also looks at the cells and stains them so that he or she can measure how many of the cells are in the act of dividing when the specimen was obtained. That's what we call the mitotic rate, the rate of cell division. We know that generally a more actively dividing tumor with a higher mitotic rate, that's a bad sign because it makes the tumor more aggressive. Now the question was, why do some GISTs have a high mitotic rate and some a low mitotic rate? Well, we don't know. We don't know. It must have something to do with the biology of those tumors, maybe mutations in other genes, maybe other biological factors. We just don't know. But we do know that it's an important diagnostic factor. Well, what was the other part of the question, Sarah, the last part of the question?
Oh, one second. It
why are
says
some high,
what causes these differences in the rates, and do subsequent tumors maintain these rates upon reoccurrence?
Okay, so I mean, it would appear that if the it would appear the mitotic index is high, it'll stay high. But of course, we don't really know that because the tumor will then be treated with imatinib, and imatinib should pretty much shut down the cell division. So the mitotic rate, we would hope, would drop basically to 0 after treatment. So the propensity would still be there, but with any luck, the measured mitotic rate would drop to basically nothing because imatinib would shut down the cell division.
Okay, next question. A lot more pouring in. What normal organ functions are diminished by TKIs, which are tyrosine kinase inhibitors?
Yeah, so that's a very important question. No drug is free of side effects, and that's true of TKIs as well. I mean, the answer is that it's a wide spectrum of side effects. Again, I go back to the fact that let me go back to say that TKIs, tyrosine kinases, are involved in a myriad of biological functions, many, many biological functions of all kinds. And so, besides cell division in GIST. And so, one would expect that tyrosine kinase inhibitors would cause a lot of side effects by interfering with other pathways in the cell besides KIT. And in fact, that's true. As I said earlier, the TKIs that are in use now, none of them were developed specifically for GIST. Most of these TKIs, well, all of them to some extent, affect other TKIs besides GIST, besides KIT, at least to some extent. And that's why we see with drugs like imatinib and sunitinib, we see a wide spectrum of side effects. And you'll hear about those on the patient chat rooms about, cardiac issues, muscle issues, hand-foot syndrome, many other side effects that people observe. Most of these are manageable. Generally, they're manageable by a skilled doctor, but it's a wide range of side effects. And to tell the truth, we don't really understand for each of these side effects what particular, we call these off-target effects. So you use imatinib to attack the target, KIT, but it also, unfortunately, will have effect on other targets, other proteins in the cell. We call those off-target effects. And we really don't have much understanding yet of which particular off-target effects are associated with which particular clinical side effects of the drug. We do hope that that's going to be less of a problem with this second generation of drugs that are more targeted for KIT.
Okay, I'm just going to jump in and encourage participants, if you're interested in side effects and side effect management, we have a platform called mysideeq.org, and that's a great way to discuss tips on how to manage side effects. So just please visit that website if you have questions about that. I really think we only have questions for one more question. Before I ask that, I know that there have been some questions that have come in about generics versus brand. I don't think David is able to answer that question, but we do have a previous webcast that we can send to you if you're interested in hearing about that, and we can always bring up a new webcast to talk about that topic as well, because we know that that is a popular topic on these chat forums.
So Sarah, one more point. Sarah, sorry to interrupt. I see somebody is asking whether they can get a copy of the slides, and the answer is yes, it's going to be put online.
Yes. We also have recorded this, and that will be available online as well. Okay, so the last question. I'm confused about therapy for mutations. If my primary tumor is exon 9 and responds to imatinib, and then I mutate to exon 13, for example, would I start a new therapy in addition to imatinib or switch therapies?
Yeah, that's a great question. It's one that I think really has to be addressed to an expert doctor, a GIST expert. I mean, in most cases, the approach has been that when resistance develops with a secondary mutation, typically what's been done is to switch from imatinib to Sutent, to a broader spectrum TKI like Sutent. It's not clear whether that's the best approach. I think the question asked whether it might make, I interpret the question as asking, would it make sense to stay on imatinib and also add another drug to deal with the secondary mutations? That's a thought that has certainly been in the air for some time. It's not easy to do because of drug interactions and side effects, but it's certainly not a crazy idea. It may be a very sensible idea, but we really don't know the answer yet. And now when we get into the question of that detail, I think that really has to go to one of our GIST expert doctors like Dr. Trent or Dr. Heinrich.
Well, thank you, David. We're out of time today, but a lot of questions poured in, and the good news is we are doing a series of these webcasts. So every month we're going to be having a different topic. Next month on April 17th, Dr. Chris Corliss is going to be talking about mutational testing, and I know that there's some questions that have come in about that. So I'll make sure that he gets some of those questions so he can address it in his presentation. And if anybody has any other questions that they didn't have a chance to have answered today or think that would be good topics for the future, feel free to email us and we will do our best to find a speaker to present on that topic. So with that, I want to thank everybody.
I've got my email address on the slide too, so they can also email me directly, sir. Yep.
Absolutely. thank you, David, so much for presenting today, and thank you to everyone who has attended today's presentation. Have a wonderful day.